Hi, my name is Braylon, and today I'm going to be going through the steps in DNA replication. So, first we have an enzyme called DNA helicase, and this enzyme is going to go in between our two strips of DNA, and it is going to open them or unzip them for the process of DNA replication to begin. And once these strips of DNA are open, we have SSBPs, or single-stranded binding proteins, that are going to attach to the outside of the parent strands to keep it open for the process of replication. So once helicase is in between our strands, in between is what we like to call the replication fork. And as DNA helicase is moving down, primase is going to be following this, and as primase moves along, it's going to be adding primers on the inside of the strands. So for the leading strand, which is going to be my top strand, it is going to place one primer along or on the inside of the parental strand. So we're going to match the bases with their correct bases, and that is our primer on the leading strand of DNA. And as our helicase and primase move along, polymerase 3 is going to attach to this primer and is going to move in a 5-3 direction down the DNA, and it is going to place with it DNA. So as it moves down, it is going to match the pairs, the bases, in the daughter strand with the bases in the parental strand. As our helicase moved down our strands of DNA, it opened up the DNA as it went along, and our DNA polymerase 3, starting off at our primer, went down in a 5-3 direction and filled the entire strand, known as the leading strand, and completed it at the very last base. And now our DNA helicase is going to, it's going to open the lagging strand. The lagging strand is also going to be open at the same time as the leading, but as it goes down, it's going to leave a primase is also following this, and primase is going to leave a primer along the lagging strand. And right here we have our primer, and as this goes down, it's going to leave multiple primers along the strand, the parental strand. So as our helicase unzipped our parental strands of DNA, primase followed along, and what primase does is adding these RNA primers onto the parental strand as a guideline for DNA polymerase 3. So since we're at the end of our lagging strand, we're going to add a primer, and so now comes our DNA polymerase 3, which is going to attach to our primers. And so with our lagging strand, the direction in which we have to go is in a 5-3. So we're going to be moving this way, but our helicase is moving in the opposite direction of our polymerase. So what our polymerase will end up having to do is, once it completes a fragment, meaning going from one primer to the next, it's going to have to jump and go to another primer and fill out that fragment of DNA. So what we're going to have to do is insert our DNA strips, which our polymerase 3 is going to be adding. So after this fragment is complete between both primers, it's going to jump from the primer here to the primer here, and it's going to continue filling in the missing fragment of DNA. Now once all of our DNA has been filled in, an enzyme known as DNA polymerase 1 is going to come in and attach to these primers and take out the primer, and with it, it's going to bring DNA pieces that replace the primers with the exact same bases. So DNA polymerase will go to every one of these primers and replace it with the exact DNA that matches the parental strand. So it's going to go to this primer and fill in the DNA. It's going to go to the last primer and fill in the missing DNA there. Now after all this is done, after DNA polymerase 1 has removed all of the primers, ligase is going to go to the places where the primers were removed and act as like an adhesive for the DNA to be attached. So where our primer was with our DNA, that piece needs to be attached. So ligase is going to bring those two pieces together so that they can stick together. So ligase goes here, ligase is going to attach here and here where our primer was, and then ligase is also going to attach where this primer was, and it's going to attach these two separate segments of DNA together.